Hello, and we're glad you've joined us for this live webinar, Prepare for Success, GMP Cell Banks as part of a staged, standardized, platform-style cell production process. I'm Susie Valdez of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots and brought to you by Thermo-Fisher, manufacturer of GMP Gibco cell therapy systems, reagents for pluripotent derived cell therapy development. For more information on our sponsor, please visit them at thermofisher.com. Now let's get started. You can submit questions to the speaker during the presentation while they're fresh on your mind. To do so, simply type them into the drop-down box located on the far left of your screen labeled Ask a Question and click the Send button. We'll answer as many questions as we have time for at the end of the presentation. If you have experienced technical problems seeing or hearing the presentation, simply click on the Support tab found at the top right of your presentation window or report your problem by typing it into the Ask a Question box located on the far left of your screen. This is an educational webinar and thus offers continuing education credits. Please click on the Continuing Education Credits tab located on the top right of your presentation window and follow the process for obtaining your credits. I now present today's speaker, Benjamin Fryer. Ben is the CEO and co-founder of Pluristyx Inc, providing GMP pre-expanded pluripotent stem cells, contract development services, and consultancy for groups and company developing cell and tissue therapies. Ben has almost 25 years of experience as a leader and scientist in large pharma, academia, and startup companies developing combination products and cell therapies. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Without further ado, let me introduce you to our presenter. Ben, you may now begin your presentation.
Thank you, Ms. Valdez. I'd like to thank Ms. Valdez from LabRoots and also Kyle Berry and Amanda Summers from Thermo-Fisher Scientific for the opportunity to present here today to all of you. So today, as was mentioned, I'll be talking about manufacturing GMP cell banks as part of a staged process and why it is so important and how that's incorporated into cell therapy manufacturing. And we'll also talk really about the critical reagents, maybe most importantly, the quality of the reagents and raw materials, also known as ancillary materials that have to go in early in the development process, and why it is so important to incorporate quality materials early on. So as an overview, I think most people that are listening to this webinar know that pluripotent stem cells, either from human embryonic stem cells or induced pluripotent stem cells, have been described in literature for at least the last 12 years for induced pluripotent stem cells, and over 20 years ago from the groundbreaking work from Jamie Thompson in the late 90s showing that human embryonic stem cells could be derived and expanded. Based on that work, there's been a lot of proof of concept studies that have been done either with human embryonic stem cells or with induced pluripotent stem cells, also just called PSCs here, pluripotent stem cells, make cells or tissues that can be transplanted into animal models of disease and can essentially cure those diseases. That includes things like blindness, diabetes, heart disease, spinal cord injuries, potentially things like Parkinson's. So of course these cures or treatments are very exciting, but translating the promise of those PSC-derived therapies to reality for patients requires really a rigorous and often expensive and definitely highly regulated manufacturing that is broken up into unit operations. So you don't just start growing cells, make your product, and ship it out. You have unit operations that define how that process is manufactured. And it all starts with making the starting material, that is to say the master cell bank, and then from that you would make working cell banks. And as part of that, you need to make sure that those are high-quality cell banks that start with high-quality starting material that will meet today's regulatory requirements, but because the process for development is so long, you need to consider that they might need to exceed the current regulatory requirements to meet tomorrow's regulatory requirements. And as part of that, integrating high-quality raw materials, ancillary materials, and in this case we'll call them ancillary raw materials, into the product development and manufacturing, starting at the very beginning with the cell source and the manufacturing of the master cell bank. In doing this, it reduces process risk, regulatory risk, and long-term it reduces the economic risk of potentially delayed product release or potentially the company or the sponsors not having enough money to continue. So where are we right now in terms of pluripotent stem cell-derived therapies? When we look at the state of the art for other biological manufacturing, that is to say antibodies, proteins for, say, enzyme replacement, or vaccines, or even virus made for other cell therapy manufacturing for things like CAR T, there's a host of cell lines that are available and that are already GMP and are ready to go. So that would be things like CHO or HEK for protein production, CHO for antibodies, HEK and viro cells for virus manufacturing. And they are essentially plug and play, meaning you can go to a contract manufacturer, and there's a host of them in the United States and Europe and Asia, and they will help you to develop a process that is defined, relatively affordable. They'll start with mostly, if not all, animal origin-free raw materials, AOF, and they will make you, starting from one of those lines that is already GMP, they will make you a seed bank, and you can pick a clone, make a master cell bank, and then proceed from there. And all of this can be routinely done from derivation as a fee-for-service. So you could have a virtual company and do it all yourself from basically an office and have a contract manufacturer do it all. That is really not the case right now with pluripotent stem cell-derived therapies that were much earlier in the development phase for that as an industry. Right now, every trial sponsor, whether it's a university, a nonprofit, or a for-profit company, currently has to develop and use their own process. And this can be inconsistent. So even if you have the same starting material, the same genetic material, whether, for example, let's say you have an H1 line you could get as a starting material from Y cell. Different expansion processes by different companies or different labs will give different downstream processing and differentiation to a product cell. And another thing to keep in mind that's a problem within the field right now is the high cost of raw materials. And the raw materials themselves are often research use only. There are a couple companies, for example, Thermo-Fisher, Stem Cell Technologies, they have some GMP or clinical grade material that is available for use early on, but many other manufacturers don't have that. So it's something to keep in mind. And when you're making these starting material, you need to make sure that you're considering the life cycle. So, for example, if you're developing the product, it can take 5 to 10 years from the time you start the development process, the early preclinical studies, till the time you get FDA approval for marketing. Then from there, you're looking at 10 to potentially up to 30 plus years for your product life cycle. So at the minimum, you're looking at 15 years of supporting manufacturing from a single bank, and ideally up to, if you're lucky, 35, 40 years for supporting the product life cycle. So the underlying master cell bank has to be designed to keep that in mind. Can you make enough without changing the bank? And the reason why changing the bank is so problematic is given the inconsistencies within pluripotent stem cells, the early bank is the foundation. It's what you build on, and you don't want to change it. The FDA requires essentially biological manufacturing to be a defined process because the end products are often hard to fully characterize. So changing, they say, the process is the product, which means if you change the process, you're essentially changing the product. And if you do that, you need to demonstrate comparability. There's 3 levels of doing that. One would be just in vitro assays. Another could be in vivo assays in an animal model. But if the FDA determines that the process has changed sufficiently and they want full proof of comparability, you could end up having to do an additional clinical trial to show comparability. So changing a process has its own internal risks, and the most expensive outcome could be redoing a clinical trial. So starting with a good master cell bank is really important. So when we look at the current state of clinical trials in regenerative medicine right now, there are 875 companies, so just over half of those are located in North America. And those companies and other universities and nonprofits are running about a total of 977 clinical trials in the regenerative medicine space. Of those, about 60 of them are with pluripotent stem cell-derived cells or tissues, and this is from clinicaltrials.gov. So there's a lot of people that are in the space, but it's, I'd say, still early, and most of those trials are in phase 1 or early phase 2. And they start, for the pluripotent stem cell-derived therapies, they start with either one of two sources. On the left, you can see embryonic stem cells. On the right would be induced pluripotent stem cells. Embryonic stem cells come from donated in vitro fertilization material that would be otherwise discarded. Usually the donors have elected to either dispose of the material or donate it. And so it's plated out, the inner cell mass grows out, you get a cell line, and that cell line can be expanded and banked, all in a laboratory under controlled conditions. And that's where human embryonic stem cells come from. One line comes from one embryo. When you're working with induced pluripotent stem cells, those can come from any nucleated cell, any somatic cell with a nucleus, and there are multiple methods to manufacture them. You can use virus, multiple viral sources, lenti, adeno, retrovirus. Those are a little bit less likely to be used for clinical grade product, but right now using plasmid or episomes is an alternative, and of course direct inoculation with protein or mRNA or a mix to induce pluripotency in an adult cell back to a pluripotent state. In the end, embryonic stem cells and induced pluripotent stem cells, if they are of good quality, they should be basically indistinguishable. It shouldn't be easy to tell where they came from. They should have the same expansion properties. The banking processes between them should be the same. So therefore, the reagents and the raw materials that are used for that process should be applicable to both types of cells. The phenotype, what you would consider pluripotency, should be the same, and they should differentiate relatively similarly, knowing that there is differences from line to line, but they should be able to form all three lineages of ectoderm, endoderm, and mesoderm. In the end, those pluripotent stem cells are what are used in the pluripotent stem cell field. So as you're taking this and you're developing your product, starting with these pluripotent stem cells, you're going to have to follow, if you're working with the FDA, their phased development scheme. It starts first with proof of concept, and this would be the same whether you're working with a drug or a small molecule or a biological or a cell therapy. You'd have to show that you've got some proof of concept, that this should have some efficacious benefit that you could extrapolate for a human being. Then you would move into your preclinical studies, which most critically involves pharmacology and toxicology. And for cell therapies, those are often mixed. For biologicals and small molecules, they can often be done separately, but for preclinical studies in cell therapy, they're often combined into the same animal model or multiple animal models. That package is then put together along with other work, including CMC and your clinical trial planning, and you file an IND, indication for a new drug, and if you get the green light and approval from the FDA, you can begin a series of escalating clinical trial phases, starting with phase 1, phase 2, and phase 3. And at the end of all that, you'll have enough data to show that it's most importantly safe, but also importantly efficacious, and then you can file for a biological licensing approval, which gives you a BLA and approval to sell your product, at which point you'd have phase 4 post-marketing surveillance. For the topic of a master cell bank, where it's probably most important to think about this, is the first 4 bullets. You have the opportunity to have what used to be called a pre-IND meeting, now called an interact meeting with the FDA, where you can get some of their early advice on how to develop your product and a few key questions that you should probably answer in advance of your pre-IND meeting. At the point when you're prepared to do your pre-IND meeting, you want to make sure that you've got an idea of what your product is going to look like. And you don't necessarily have to have a master cell bank, a GMP master cell bank at this point, but you need to have a good idea of how you're going to make it, because at this point you're going to get approval from the FDA for what you believe your acceptable toxicology and pharmacology animal models are. And this is critical because this is a very expensive process. It could cost hundreds of thousands of dollars to run these studies, and they have something that money can't buy, which is time embedded in them. So if you're going to use a pluripotent stem cell-derived therapy, you're going to have to do some sort of tumor study or teratoma formation study. and this is often to the life of the animal. So that can be anywhere between 6 to 12 months of waiting to see if animals develop tumors. And that is required to start with your master cell bank. The FDA, as you can see the link below, they require the manufacturing process of the cellular product used in the preclinical study should be as similar to the intended clinical product as possible, meaning you should start with your master cell bank that you're going to use for your clinical grade manufacturing. So you can't do those studies until you have that master cell bank. And so you can see how early on you're locked in. You haven't even filed your IND, and you need to have locked in one of your manufacturing processes. Once you've got all those pharmacology and toxicology studies, you'll file your IND, and you'll be able to move into manufacturing for your phase 1 clinical trial. The FDA has an explicit and implicit expectation that you will improve your manufacturing process from your phase 1 clinical trials through to the end of your phase 2B clinical trials. At the beginning, before you begin phase 3, you have to have locked your manufacturing process, but that allows you some downstream improvements. But the one thing that it's very difficult to improve is the master cell bank. Although you can improve lots of other things by the end of phase 2, it's really hard to make any changes to the master cell bank because that essentially introduces comparability risk. So when you think about this, if you kind of like break it up into what I've called process units or unit operations, when you're doing the research work, it really is almost seamless. You have your starting material, might be a cell bank or cell line that's floating around the lab. Maybe researchers, grad students, postdocs have traded the line around. Everybody's got their own so-called bank or master cell bank that they use. Passage number may not be well tracked. The processing is not necessarily consistent, but everybody seems to get a pretty good result, which is why you're moving forward and trying to raise money to make this into a clinical grade process. You expand the cells, and you might seed them a little lighter one day or a little heavier another day to make sure that you don't have to come in and pass cells on a Saturday or a Sunday. And at the end, you have a differentiation process, which works pretty well in a small scale. And then those are put into either just in vitro studies or they're put into in vivo studies in your animal model. And it's relatively seamless. It's often done by 1, maybe 2 or 3 people, but it's a small team at a minimum, or at a maximum, I should say. When you move into clinical grade manufacturing, that early part, that starting material, is broken up really into 3 unit operations, and it's very tightly regulated. And ideally, it should be locked early in development. That's derivation. So if it's an embryonic stem cell, that would be anywhere from getting the donation of the blastocyst or the embryo. In the case of an induced pluripotent stem cell, it would be how you're manufacturing the iPS line from the very beginning. What are the materials you use to derive that induced pluripotent stem cell? The expansion process then can be broken up into a seed material and frozen, and you can characterize and pick your starting material. Once you've picked it, then you make your master cell bank, and all of this is relatively tightly locked, and this should all be done with high-quality ancillary materials, high-quality raw materials, so that you don't have to go back and repeat this later on if there's any questions about the quality of those materials. Once you move into the later stages shown in pink on the right, which would be making the working cell bank, then thawing a working cell bank, which could contain anywhere between 1 and 5 million cells in a vial. You'll expand that up, and the seed train really just means you're growing up lots of pluripotent stem cells for your eventual differentiation process. So you might need to make hundreds of millions of cells or even potentially billions of pluripotent stem cells. At that point, you're ready to differentiate those cells into your product. And then you have finish and fill, whether it's cold stored and delivered directly to the clinic, or you have cryopreservation somewhere in that process, and then that's delivered to the patient or to the point of care. All that in pink, you could potentially make process changes and optimization prior to process lock in advance of your registration trial, which is usually phase 3. But if you have, say, an orphan designation or an advanced regenerative medicine application that's approved, that could potentially be phase 2B or something like that. But you need to potentially optimize the process from the point of the master cell bank derivation, and you have room and time for that. You don't necessarily have it for the banking process of the master cell bank. And so if we look at really what goes into the workflow that's used for making a master cell bank for an intended clinical product, it has to be made under GMP, good manufacturing practice. And this starts first and foremost by where you're going to do it. It has to be done in a clean environment, and that's because most of the operations for making a master cell bank are open. Open would be just kind of your standard 6-well plate or a T-flask. You've got pipettings that are done open. In more advanced biological processing, often things are closed and considered to be sterile. That can be done in a room environment, but for any sort of aseptic processing, it has to be done in a biosafety cabinet, which would be an ISO 5 environment, inside of a clean room, which would be an ISO 7 environment. That room and that biosafety cabinet have to have HEPA-filtered air, and the environment is monitored by things like settling plates, active particle monitors, and culture plates for monitoring for both just total particle burden and colony-forming units. Any sort of instruments that are used in that environment have to be qualified for use and for work by installation, operational, and performance qualification. Any of the operators and verifiers who work in that environment have to be gowning qualified, qualified to be in the room, and also qualified to perform the unit operations that are going to be done. And all of that has to be captured within a batch record. That is either paper or an electronic document that verifies and tracks all the work that was done and so that there's a record of what was done. And there also has to be a system to track if any sort of deviations occur, if anything that was unplanned or not captured within the batch record, to determine on a risk analysis if it impacted the quality of the product. Anything that comes in and is used in that environment has to have traceability. So that would be any sort of raw materials or excipients that are going to come in. And that means the product has to come in with a certificate of analysis, a C of A. And they're held in the warehouse in a quarantine area until they're approved for use. And that provides incoming goods control for anything that's coming in. And one thing that's not on the slide, but keep in mind, is that any sort of supplier is often audited, or usually should be audited, either with a paper audit indicating that there's some quality supply chain system used by and manufacturing system used by the supplier, or potentially, depending on the risk of the incoming product or the phase of the manufacturing, an on-site audit by quality assurance from the sponsor. Another thing to keep in mind is that the donor tissue, the cell itself, whether it's a human embryonic stem cell or an induced pluripotent stem cell, you need to make sure that you have fully consented donor release of that material. And this is critical because in today's day and age where gene sequencing and identity is so easily performed, you could quite easily, assuming that everything's been anonymized and randomized and you have fully donor consent, that donor could be traced. And very easily. And so you could imagine how family might want to make sure that they were compensated if the donor consent wasn't fully in place. And then finally, so assuming you have all those things, you've got a fully consented donor, including commercial rights to use those cells, all raw material traceability, clean room, et cetera. You've made your bank. Now the bank has to be released based on assays for 4 or more things. These are the key 4: safety, identity, purity, and viability. And so when you're making a master cell bank and also a working cell bank, these criteria hold for both form. You'll have to test for phenotype and identity, purity of the bank, the karyotype, meaning it has to be genetically stable. It has to have only one donor, and that donor has to be human. There's multiple ways to do that, but right now for human pluripotent stem cells, probably the best way to do that would be to use short-10 and repeat. You test for sterility, and that would include mycoplasma, fungus, and bacterial growth, and then also endotoxin, which can be either a result of any sort of risk from your raw materials or bacterial growth within the product. Then the product, before it's frozen and after it's frozen, you would check for viability, usually by a qualified method, which could include things like dye exclusion. And some of those are old tested methods like trypan blue, ethidium bromide, or propyridium iodide. There are a lot of other methods, but in the FDA requirement is that the bank after thaw is greater than 70% viable. And then finally, and maybe most important, you need to make sure that the bank functions as you would want it to in your process, and that would be sponsor-specific differentiation to whatever it is your fate of interest. And so that's for releasing a master or working cell bank. A master bank has a deeper dive. So when you're releasing it, you'll also have to look for any sort of viral potential contaminant or infection in that line. There's qPCR assays for the top ones, including things like HIV. There's RT-PCR for RNA viruses like HCV. You have then and of course, because they're PCR, they are primer-based, so you know the sequence. But then there's also the risk that there could be viruses that you don't even know about. So that includes adventitious viral contaminants for non-specific reverse transcriptase. And then there's in vitro and in vivo assays that are intended to determine if there's any virus in the bank. So you would co-culture with MRC5, Vero, or A549 cells and do an in vitro assay. You'd also do an in vivo assay. There's direct inoculation of eggs or rodents to look for viral contaminants. Also, if your product has ever come in contact with any sort of bovine material, you'd also have to potentially do bovine virus detection. And if you have used anything that was porcine, like trypsin, derived from pig gut, or you've co-cultured them with mouse feeders, you'd have to do porcine or murine virus panels. So it's really important that you have, if you can get animal origin free, recombinant and human materials only, then you will reduce the risk of having to do any additional viral testing. It's also really important that you understand the material supply chain of your suppliers. How did they make those materials? Because that's where additional risk comes in. Any of that raw material contact puts that master cell bank, and therefore your entire process and potentially company at risk if you don't have high quality materials coming in. So I've given you a lot of tables. I'll give you a picture just to kind of describe this. So like I said, we're looking at unit operations, a cell banking process. You start at unit 1, and that's really making your seed material, your seed bank. And with the embryonic stem cell, it starts with an embryo or a blastocyst. It's cultured out, and in the case of induced pluripotent stem cells, it'd be your donor's somatic cells that are induced back to pluripotency. You might either bulk passage or single cell clone, and then you would have potentially one or more seed banks. You could take those banks, and before you move on to unit operation 2, which is the actual master cell bank, you would take that material and you would test it. You'd want to make sure that it was genetically stable. If it's an iPS cell, you didn't have any, say, CMYC or OCT4 incorporation into the wrong places. You'd want to make sure that it had a nice population doubling time. It didn't grow too fast. It didn't grow too slow. It was a nice, stable line, and probably most importantly, if you're making a product, that it differentiates to the fate that you want. So now you've got a seed bank, and let's say that's somewhere between 5 and 20 vials. You're going to take one of those vials, and you're going to return to the GMP facility, and you're going to make your master cell bank. And that's going to take somewhere between, say, 2 to 5 passages. You start in a small, either a 6-well plate or a small tea flask. You can grow that out into a cell factory or a cell stack, and or multiple tea flasks, and you'll have enough cells to make 100 or more vials of a master cell bank. That can then be used, thawed and used to make a working cell bank, and nearly the same process as the master cell bank. And in this case, when we talk about the lifeline or the life cycle of the product, 100 vials of a master cell bank can make about 100,000 total product lots, because each vial of master cell bank can make about 100 vials or more of working cell bank. So you just multiply that out, assuming that each working cell bank vial can make a product lot. That's 10,000 possible product lots from relatively small banks. And assuming you do 2 production runs a month or just a little bit more, that's about 25 production runs a year, which would be a really high number. Normally you'd want to do a lot less than that, but let's say you're really running full gun and full bore, and you've got 25 product runs, product lots being manufactured a year. Off of this simple scheme, that would be enough for 40 years of your product life cycle. So it's enough to support a long-term manufacturing process. So just in summary, you've got your seed material process, starting either with an embryo or a small amount of donor cells. You're going to make somewhere less than, say, 20 vials. You're going to check those vials in that bank for, do they work in your process? Are they the right material? Then you're going to have to release them. You're going to want to know, in the case of an iPS cell, if you have any sort of residual virus or plasmid incorporation or episomes left in the cytoplasm. You're going to check to make sure, are they pluripotent, are they genetically stable, and of course, are they clean, are they sterile, and do they function in your process? Then you pick one, you move into your master cell bank, and now you're doing this much larger release specification, compendial release for sterility, endotoxin, mycoplasma, checking for their pluripotency. Are they the right identity? Are they genetically stable? Do they function? And in the case of iPS cells, do they have any sort of insertions that you don't want? And then a working cell bank is a subset of those release specifications. And in each case, you don't have to make a lot of vials. I mean, this isn't a large production process. The thing about it is it takes a lot of time. And as you can see here on the third or fourth line, time to completion, it could take 14 to 21 days or more for the seed material, 20 plus days, potentially, or around 20 days for a master cell bank, and another, say, 15 to 20 days for your working cell bank. If you add that all up, you're looking at just in time alone spent in a GMP facility of about 7 to 8 weeks. And so this is expensive. GMP facilities are not cheap. Operations are not cheap. And so you need to plan these out very carefully in advance because you don't want to change the master cell bank. The cost, even if you do it early on, is quite expensive. A GMP suite with reagents and labor. And I think these are on low-end numbers. They're tens of thousands of dollars. The actual bank release testing for the tables I showed previously, those are hundreds of thousands of dollars. So they're quite expensive. And the time is something that money just can't buy time. Just to generate and test those cell banks can take 9 months or longer, including the tumor studies that are required for your toxicology studies to sponsor and support an IND filing. And so the risk to the sponsor if the cell bank isn't done properly are, if you change it, cell banks by nature are inherently variable in performance. You could end up having to go back and re-optimize your manufacturing for your differentiation, and that cost could be millions of dollars. The additional cost could be in comparability requirements that any regulatory agency might require. And so if you imagine that you've got a product that could be a blockbuster drug, meaning it makes $1 billion a year or more, and you end up taking a 9 or 12-month delay because you had to remake a master cell bank, that's $1 billion in cost. It's money that actually hasn't been realized, but it could be the difference between success and failure of a company, especially if you have a competitor that you're neck and neck with and they get there first. You could end up losing completely. So just to review the risks to the master cell bank, and on the slide after this is really how to mitigate these risks. You want to make sure you have proper donor consent for the incoming raw material, that is the cell itself. You want to make sure that any sort of raw materials or ancillary materials that are used in making your bank are of high quality. So if you're going to use feeder cells, I'd recommend GMP human feeder cells. If you're going to use, I would recommend against mouse feeder cells. You can use them. For example, H1 and H9 have been used for human clinical trials, but they are not. They're considered xenogenic lines, meaning that they've been in contact with another species' cells, and so it's not something that you'd want to do if you can avoid it. But it's not a game stopper. If you're using any sort of bovine component in your process, FBS or bovine albumin, you want to make sure it comes from a qualified herd, meaning a USDA-approved herd that has a certificate of origin. And this is because of prion disease. There is no assay for it. The only way to prevent it in your manufacturing is to start with high-quality material. And if you can't prove that later on, that could be a game stopper. So TSE has a real risk to your process and your product. The media, depending on how it's made, there's in warehoused, actual mice in the warehouse can transmit virus to the components, and so you want to use media from a reputable vendor who actually tests for that. Your growth factors should be all ideally human recombinant and, if possible, animal origin-free, and that includes for pluripotent media, things like insulin, BFGF, and TGF-beta, and other growth factors that can be in the media. Your small molecules, if possible, should be filtered, and your DMSO should be of pharma grade. Cell culture plastic, usually if you're working from something like Nalgene Nunc or Corning, you're going to get a very high-quality product, but some people use cheaper sources. I'd recommend against it. Go with a reputable, old, and trusted vendor. If you're using a coating, which most pluripotent stem cells require a coating, avoid Matrigel if you can. That's a really RUO product. You want to go with something that is manufactured fit for purpose, and that would be a recombinant product or a synthetic product like RGD or recombinant laminin. And for any sort of human donor materials, you want to make sure that you have a certificate of analysis that they are clean for any sort of viruses. And just keep in mind that the real risk here is two things: transmissible spongiform encephalopathy, the prion disease, and viral contamination. And you can test for viral contamination later on, but you can't test for TSE. So you need to make sure that anything that's made, any product that's made with bovine source in your supply chain, or that is a bovine source material that you're using directly, has come from a USDA certified herd or New Zealand, Australia herd. So in terms of how do you mitigate this, the USP has very clear guidance on this. The 1043 was recently issued, I think a couple of years back, and it talks about how to incorporate ancillary materials for cell, gene, and tissue engineered products into your manufacturing. And they do this by providing a 4-tier table system. So Tier 1 is the lowest risk, Tier 4 is the highest risk, and Tier 4 really includes a lot of the things I'm talking about previously, like bovine-derived materials, any sort of xeno product, anything that's not intended for potentially incorporation into an infusible or directly treatable product. Things that are for diagnostics, say, or for research would be in Tier 3 or Tier 4. And they provide guidance for qualifying the use of materials. So if a material is, say, research use only, but you have to use it because there is no GMP or cell therapy grade product, they provide guidance for qualifying that material to use in manufacturing your cell, gene, or tissue engineered product. And that is key because, early on, the FDA and other regulatory agencies recognize that there is a risk. You can't always get a GMP reagent if you're pushing the envelope and trying to make a new therapy. They do expect that you work with a supplier that is committed to improving their quality systems in parallel as you advance through your phased development to commercial manufacturing for human use products. And that means you really want to make sure from the very beginning you work to integrate high quality raw materials and ancillary materials into your manufacturing process from the very first seed bank onward. And that means, if you can, use GMP or animal origin-free products as soon as possible, especially for your cell banking. And source all your raw materials, if possible, from suppliers experienced in supporting cell therapy development. Ideally, these should be stable, long-term partners because they're going to have to commit to supplying that product to you as long as you're making your product. And if they do so, that will reduce your risk of potential disruptions, things like back orders. I know if you're in a lab and something's backordered 6 weeks, it's a hassle. If you've got a GMP suite set up and you've got operators and you've paid for that time and you're backordered on a critical reagent, you have to pay for that time. That can cost a lot of money. Product changes should be clearly communicated to you if you're using them, and you don't want to have to worry about any sort of discontinued products because that can, of course, create great risk in your manufacturing system. And you want to identify, if possible, more than one source for your raw materials. Ideally, at least 2 suppliers. Avoid sole supplier products if possible. I know that's not always possible, but that's something to keep in mind. It just reduces your risk as a manufacturer or as a sponsor. So in summary, I think I've hopefully communicated how important it is from seed material on through your master cell bank all the way through to end product manufacturing, how important it is, especially at the beginning, to use high-quality raw materials from an established and reputable vendor as early as possible, because those raw material risks extend throughout the supply chain, and they put the entire project and product at risk if they're not dealt with. You want to look and future-forward planning. You want to know where you're going to be, where you want to be, knowing that it's going to take many years, and you want to anticipate increased regulatory stringency over time for your product. And so you don't just want to meet today's standards. You want to know that tomorrow's standards are going to be a little higher, and you want to try and meet those in advance so that you can at least pass the bar for tomorrow, even though you greatly exceed it today. And all of this, if you do this and put this in place now, you can avoid the loss of time, money, and potentially catastrophic risk to your product or your company by generating a high-quality seed and master cell banks in the beginning that can sustain the product throughout its life cycle. So My last slide, just a little bit about Pluristyx. I won't talk too much about it, but I'd like to thank everybody who has come today and watched and listened to this talk. And if you have any questions, I'm happy to take any questions you have at this time. Thank you.
And thank you, Ben, for your presentation. A quick reminder for our audience on how to submit questions. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Before I ask the first question, we have a question for you. Have you worked in a GMP environment before? This question will now pop up on your screen, and thanks for your participation. So let's take a look at our first questions coming in from our audience. Ben, can you make a cell line GMP if it wasn't originally generated in a GMP environment?
Yeah, it's an excellent question. So this is a common question that's asked, and I think the answer right now, I think, is yes. So for example, the H9 line that is available from YCell was made under research conditions, so that would have been considered a research master cell bank. That bank has been used essentially as the seed bank, so as a unit operation, and that research master cell bank becomes the so-called seed material. A vial of that is then transferred into a GMP environment. An entire batch record and process is developed around that, and then it's manufactured and released. And there's essentially a risk assessment that has to be done on all the materials that were used to make that line in the beginning. So it's kind of a way of kind of figuring out what other additional viral panels you should look for. I guess the biggest risk with any sort of with doing something in that manner is that you need to make sure you have early passage cells, and it's actually not given that the FDA or other regulatory agencies will accept it by the time you get to filing your BLA. So you might be able to make your product early on, but you might be essentially pigeonholed into a very small indication. So for example, if you wanted to go for, show that you were making, let's say, neurons, and you wanted to treat a specific type of blindness, you might only be able to use that product for that specific type of blindness. To be able to add on indications or other therapeutic areas, you might not be able to do it because they would consider that product to be too high of risk, and so you'd end up basically having to restart the process. So I guess the simple answer is yes. It's possible to take a research bank and make it into a GMP master cell bank right now. But I think if you can afford it, if it's possible, it's better to make the master cell bank from a GMP source from the very, very beginning.
Thank you, Ben. And is there a difference between a research master cell bank and a GMP master cell bank? And can a research bank be converted to GMP bank during development?
I would say probably you can. Like I said, it's possible to convert from one bank to another. I think that the cost is really the issue in terms of risk down the line. And so I would recommend, if possible, especially if you're working with an iPSC, start with a GMP seed material and a GMP master cell bank. Embryonic stem cells a little bit harder, but it is possible to take a research master cell bank, make that your seed material, and make a GMP master cell bank later on.
Thank you, Ben. And one audience member would like to know, to your slide 18 point, how important is it to you, quality by design?
So quality by design essentially is asking the question beforehand, how, testing the parameters of your process. So it's a little bit easier to do with a master cell bank. Really what you're looking for is population doublings, and essentially you're controlling for risks and stresses in the process early on. I think it's important to use, but I don't know exactly how much work is required to do quality by design early on. I think having the good master cell bank probably feeds more directly into quality by design later on in the process, so that you have a starting consistent process and product that the bank that you can use for manufacturing later on.
Thank you, Ben. And why do you need a GMP cell line to make study material for pre-IND-enabling studies?
So the risk, so the request in a pre-IND, in an IND submission is that you have to have shown that your material is, number one, and most importantly, it's safe. And so the safety question for a stem cell, any sort of cell which has the ability to grow and form all the other tissues, is that it could form a tumor. And so the question is that, you have a master cell bank. That master cell bank is where it's essentially the starting point for all things. So if you generate different master cell banks, the potential is that one of them could have a different risk for tumors or for ectopic tissue formation. So if you do your combination pharmacology, toxicology studies with your master cell banks, and they're not the one that you're going to use in patients, the FDA or some other regulatory agency is going to wonder why or what the other risk is. And so this is probably the biggest problem with trying to change master cell banks. You'll end up having to redo that study. So you could potentially use one master cell bank if it's GMP for your pre-IND enabling studies, but if you want to switch to another bank, you'll probably end up having to do those teratoma toxicology studies again, and that just cuts down. It's a time cost, so you could end up having to repeat human clinical trial studies. So I think it's really critical to get that done first.
Thank you, and thank you to our audience members for their questions that are coming in. Then our next question is it important to have a MCB if the cells being used are from the source they are going back into? For example, from patient A back to patient A and no one else?
Yeah, so this would be essentially almost like the autologous model of cells. So if there's this question of minimal manipulation, so for example, if you're pulling cells out and you're going to put them right back in, so for example, if you're banking blood from somebody for a surgery, and you want to make sure that they have a blood supply that you could infuse back into them, that's considered minimal manipulation, and you won't have to make a master cell bank of that. But if you're going to expand those cells and you're going to change them, you're going to end up having to do proper release on that product, and that release panel is expensive. I guess it really depends on the product. So, for example, if you're making a pluripotent stem cell derived product, you're going to have to make a master cell bank, and you're going to have to release the master cell bank. And the real question there is the risk to the patient, and the risk comes from the raw materials. So virus and sterility can be a problem based on whatever you're doing to expand those cells and to change them. So you'll need a master cell bank. The full release panel may not be to be so extensive, but you will need a master cell bank.
Thank you. And Ben, does the master cell bank strategy change if the pluripotent stem cell is genetically modified? Example, expresses a transgene or has a gene edit?
I love this question because it's a question I've asked before. So I think that, let me just preface my answer by saying I don't think my answer is the only answer, and I'm not sure that the FDA would agree, but I'll give you my opinion on it. And that is, you'd want to start with a master cell bank that was not edited, and then you would make essentially working banks that would be genetically modified. But all of that then has to be done in a GMP environment, and you have to fully characterize the working cell bank that's genetically modified. So you may not have to redo all the viral testing, but you will have to do genetic stability on that working cell bank.
Our next question is, we have a clean room that is not GMP. Can we manufacture our material there and use it for clinically representative pre-IND studies?
So for a clean room that's not GMP, I think if you're making your working cell banks or so-called GLP material that follows the supply chain and the batch records and the process that you use for making the end product, you can manufacture material for your pre-IND studies that's not in a GMP environment. However, the master cell bank, that does have to be GMP. So at some point, you're going to have to make a break and make that GMP master cell bank. knowing that you can take some of those vials back to a research lab, which is clean but not GMP, and make essentially development or research working cell banks, and under GLP or sufficient quality to meet regulators' requirements, you can make your end material, but you can't skip the GMP master cell bank. So I guess when you get into final or actual phase 1 manufacturing for patients, everything, soup to nuts, has to be done in a GMP environment. But if you're making material for pre-IND enabling studies, preclinical and animal studies, that has to start from a GMP master cell bank, but you could make it under GLP in a less clean or less controlled environment for IND enabling studies.
Thank you, Ben, and we appreciate the live participation from our audience. We have time for a few more questions. Our next question is, what is the difference in the cost of cell culture for GMP products and for laboratory purposes?
Probably the biggest cost is oftentimes research use only, or RUO. I didn't spell that out in the talk. RUO just means research use only. And a GMP product, often there's very little, maybe no difference in how they're made. The difference is in the amount of labor and time and documentation that's required to differentiate between a GMP product or a cell therapy grade product and a research use only product. So that's why there's so much more cost. It's in the human side, and the liability that is involved in that for the supplier. So GMP reagents are just by nature more expensive because there's a lot more traceability that goes into that. The testing's more extensive, things like that. So when you're working in a GMP environment, the same kind of thing. The lab will look the same. It'll have an incubator. It'll have a biosafety cabinet. It operates like any other lab would, except for the fact that you have to have controlled entry, controlled exit. You have to have traceability in the environment. You have to have traceability on operators, traceability on all the machines. And so just the expense of keeping that record and keeping that clean room is what causes the GMP prices to go up. I guess in the short end, the difference is really traceability and human labor that's required for that, and therefore that is what increases the cost between GMP and non-GMP.
Thank you for that answer. Our next question, which molecular assays do you suggest for testing genetic stability of the MCB?
Well, right now, the assay, which is, and it's not molecular, it's gross morphology essentially, is G-band. G-band is considered the gold standard, and it's what's used to test for genetic stability. So you'll look at, say, 30 to 50 cells, and you should see the karyotype should be normal, stable. A lot of groups are now starting to add in newer technologies. You've got whole genome sequencing. You've got integrations, looking to see if there's any sort of, especially for pluripotent stem cells, if there's integration of your pluripotent genes into the genome of the cell of interest. I mean, I think there's a fair amount of assays that you can use to give yourself comfort, to give your investors comfort, to give your patients and your physicians comfort that you have a stable line. But right now, the requirement would be karyotype.
Thank you, Ben. And we have time for one more question. Our final question is, what are the consequences to not using a GMP master cell bank?
Your IND won't be approved. So you'll do a lot of work, and you won't be able to begin clinical trials.
Ben, do you have any closing remarks that you'd like to share with the audience now?
No, I guess yes, I do. So I'd just like to thank everybody who came and participated, listened to our talk. If you have any questions you'd like to follow up with, you can find us at our website, pluristyx.com. And I look forward to hopefully meeting some of you in person, and if we can do anything to help you, we'd love to help. And I'd like to also thank Thermo-Fisher and LabRoots for the time and attention today, and for inviting me to present. This has really been fun. Thank you. And
before I go, I'd like to also thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. The webcast can be viewed on demand through August 2019. LabRoots will alert you via email when it's available for replay. We encourage you to share this email with your colleagues who may have missed today's live event. A poll will now pop up on your screen, and your answers and your suggestions will help improve our webinar program, and they are greatly appreciated. Until next time, bye-bye.